Today, we'll see how soil microorganisms break down organic matter. You'll need 2 soil samples, one left as it is, and one sterilized by heating it on a hot plate for about 20 minutes, 2 petri dishes, water, and small pieces of filter paper. Put the non-sterilized soil in the first petri dish, and after the sterilized soil has cooled, put it in the second dish. Place a small piece of filtered paper on top of the soil in each petri dish. Next, lightly moisten both dishes with a little water. Then close them. Keep the petri dishes in a warm place and check on them regularly for 10 days. Once 10 days have passed, compare the 2 petri dishes. In the sterilized soil, the paper will remain practically unchanged. In the non-sterilized soil, the paper will be partly or fully degraded. This degradation happens because microorganisms in the living soil, like bacteria and fungi, secrete enzymes that break down organic matter. This shows how important microorganisms are for recycling matter in the soil. To go further, you can repeat the experiment with wetter or drier soil to see how moisture affects the rate of decomposition.